Of you. In this video, we'll be discussing the nucleic acids chemistry, their structure and composition, what are mononucleotides, and the difference between the nucleosides and the nucleotides. So the nucleic acids were first discovered in pus cells and salmon sperm cells by Friedrich Mischer. He named them nuclein because of the source. And then later on, it was found that they are acidic in nature. So Richard Altman named them nucleic acids. Nucleic acids are important biomolecules because they are responsible for the existence of living beings as they make up the molecular repositories in terms of the DNA and RNA, which can function as the storehouse of genetic information in prokaryotes as well as eukaryotic cells. So they not only act as repositories but also transmit this genetic information down the line through central dogma, result in the formation of the proteins which do the function and result in the existence of these cells. So what they are actually are the biopolymers of a repeating unit, which is a mononucleotide, which is repeated over every time, resulting in the formation of the long run of chain, which is called as biopolymer. And these mononucleotides are together bound with each other by 3 prime 5 prime bridges, which are made of phosphate. So these building blocks, the mononucleotides, which make up a polymer, that polymer can be RNA or DNA, hence they are the building blocks of nucleic acids. They are analogous to amino acids role in proteins. Just like in proteins, which are made up of polymers of amino acids, nucleic acids are the polymers of mononucleotides. Further, there are certain mononucleotides which can serve as energy currency, like that of an ATP as well as GTP. Allosteric effectors are some of them which are needed for the functioning of the enzymes like ATP, GTP, CGMP, or cyclic AMP. Similarly, they make up certain cofactors like that of NAD, nicotinamide, adenine dinucleotide. They can function as oxido-reducing agents as well. For example, NADH. They serve as activated intermediates in numerous biosynthetic pathways like that of an glucose pathway or HMP shunt or biosynthesis of fatty acids. And they also act as an mediators for the cellular processes as second messengers, like in case of cyclic AMP, which serves as an second messenger to various hormones in signal transducing. So their composition is actually of 5 different elements, that is carbon, hydrogen, oxygen, nitrogen, and phosphorus. So nucleic acids are made up of these 5 different kinds of elements. As I already said, that structurally nucleic acids are the polymers of mononucleotides. This polymer is possible only when they are linked. The mononucleotides are linked with the help of the bridges, which are phosphodiester bridges. The phosphate of the one nucleotide joins with the another nucleotide to result in the formation of this bridge. So each mononucleotide, when read as a separate entity, it is made up of 3 different components. One is the sugar base, another is the phosphate ion, and third is the nitrogenous base, as you can see in the accompanying picture. Depending upon whether it is a DNA and RNA, however, both of them are made up of these 3 units. Only the difference lies in the sugar base, which I will tell you later on. So a mononucleotide, whether present in the DNA or RNA, has 3 different components in it. The sugar component of the nucleotides is 5-carbon aldose, pentose, which is present in furanose form, as you can see in the accompanying picture, the 5-carbon unit of the sugar. The difference between the 2 different nucleic acids, the deoxyribonucleic acid and the ribonucleic acid, is of the sugar. In RNA, it is ribose type, and in DNA, it is deoxyribose type. The difference between the two is in the carbon 2 of the ribose and deoxyribose. In deoxyribose, the oxygen at the carbon 2 is not present, while as for the ribose, the carbon 2 contains the OH group in contrast to the deoxy one, hence the name deoxy. So the nucleic acid which contains the deoxyribose type of an sugar is always deoxyribonucleic acid, DNA. Another important component of the nucleic acid is the phosphate. In soluble form, it is present as H3PO4. So it has three monovalent hydroxyl groups. So it can form three different bonds. But here in nucleic acid, it forms two bonds. One bond is formed between the 3' hydroxyl group of the ribose or deoxyribose and the phosphate, and the another bond is between the 5' end of this sugar with the 3' end of the another mononucleotide. So it links the 5' of one mononucleotide to the 3' of another mononucleotide to result in the formation of a run of mononucleotides, hence polymerization. And this linkage is called as 3' 5' linkage. The third important constituent of the mononucleotides is the nitrogenous base or nitrogen base. It is of two different types derived from the pyrimidines or purines. The pyrimidines, as you can see in the accompanying picture, is here. It's a single heterocyclic ring molecule which is present in the three different forms in the nucleic acids, and then purine is containing a heterocyclic double ring, consists of an imidazole ring and a pyrimidine ring, and it is present in two different forms in nucleic acids. So the purine derivative which is present in the nucleic acids are adenine and guanine, as you can see here, adenine and guanine, and the pyrimidine derivatives present in the nucleic acids are thymine, cytosine and uracil, as seen in here. Chemically, adenine can be said as 6-aminopurine and is usually found in both DNA and RNA. Guanine is called as 2-amino-6-oxypurine and is found in both DNA and RNA. As you can see in the picture here, both purines are found in both DNA as well as RNA molecules. While as for the cases of the pyrimidine, there are three different pyrimides present in the nucleic acids: cytosine, thymine, and uracil. Cytosine is 4-amino-2-oxypyrimidine and is found in DNA as well as RNA. Thymine is 5-methyl-2,4-dioxypyrimidine. It is exclusively found in DNA only. It cannot be found in RNA. While as in turn of the thymine, uracil is present in the RNA, but it is not present in the DNA. Uracil is 2,4-dioxypyrimidine. Their structures are here, as you can see in the accompanying picture. You will see the uracil is present exclusively in the RNA, while as thymine is present exclusively in the DNA, hence giving them distinct identity, apart from the difference in the ribose and deoxyribose. Deoxyribose is present in DNA, whereas ribose is present in RNA, as I already told you. So what is a nucleoside? Nucleoside is actually a combination of two different things, the nitrogen base and the ribose. When both are linking together at a carbon number 1 of the sugar moiety, then the association is called as a nucleoside. So the association between a nitrogenous base and ribose happens with the help of a bond. That bond is called as glycosidic bond. And nucleosides usually contain either the ribose as sugar or as the deoxyribose as sugar. If it is ribose, then it's called as a nucleoside will be called as ribonucleoside, and if the sugar to which the nitrogen base is attached is a deoxyribose, then the nucleoside will be called as the deoxyribonucleoside. So deoxyribonucleoside contains the deoxyribose, and ribonucleoside will contain a ribose sugar attached to the nitrogen base with the help of a bond, that is a glycosidic bond, as you can see in the picture here. The bond between the nitrogen base N1 and the C1 of the sugar. So it is a ribonucleoside. Similarly, here you can see the bond between the purine N9 and the bond between the C1 of the sugar. It will result in the ribonucleoside, which is a purine type. So the bond will always be, the glycosic bond will always be between the C1 and N9 of purine and C1 and N1 of the pyrimidine. So pyrimides are connected to the sugar moiety through N1, C1 and N1, and purines will remain connected to the sugar via N9, that is C1 and 9. So this is the bond, what bond? Glycosidic bond, and this glycosidic bond is always in a beta configuration. So far we know that nucleic acids are the polymers of the mononucleotides, which are repeated and linked with the help of a bridge called as the phosphodiester bonds. So a single nucleotide will be the one which is composed of all three components which we just went through. That is sugar, phosphate and nitrogen base. These are also referred to as sometimes as the phosphoric acid esters of nucleosides. That means that they are made of nucleosides by the addition of a phosphoric acid to their one end. This usually is the 5' end, as you can see in the picture. The phosphate, when bound to the sugar at the 5' end, and nitrogen base at the C1 end, this whole moiety will be called as a nucleotide, and usually they are existing in nature in both monomer form or as a polymer linked together by the help of an phosphodiester bridges. In isolated form, if they are present, the phosphate will always be bound to the 5' and that is carbon 5 of the um, sugar moiety. Whether that sugar moiety is a ribose or deoxyribose does not matter. So as you can see in the picture, there are three different kinds of nucleotides which are existing in the usually nature. Adenosine 5' monophosphate or AMP, sometimes also called similarly guanosine or GMP and CAMP. So when the moiety or nucleotide is AMP, it means that it contains one phosphate bound to a sugar and contains nitrogen base as adenosine A. Similarly, if it is GMP, then the nitrogen base will be the guanine and the sugar will be either of the two, and the phosphate will be bound always to the 5' end. This is a mononucleotide. Then there is another called as dinucleotide and trinucleotide, which I will tell you later. So mononucleotide contains only one phosphate, always bound to the 5' end of the sugar moiety. So some of the mononucleotides which exist as monomers in the cell and do the varied functions are necessary sometimes for the existence. Specifically, some of them, like for example, the prime one is the ATP, which acts as the energy currency of the cell, and it determines the cellular capability of the cell for doing a certain function, and for its existence it is necessary. uh, Similarly, there is a GTP which almost does the similar kind of a function as that of ATP. It provides the energy input specifically for the translation process when proteins are being synthesized by the ribosomes. So ribosomes depend upon GTP as well. CAMP, which acts as a foremost cellular messenger for many kinds of hormones. So the hormones mediate their functions with the help of this CAMP cascading mechanism. GAMP also similar kind of an second messenger, which does the function of nitric oxide signal transduction, and cellular reducing agents like NAD and NADPH, which are necessary for the uh, oxidation of the glucose, proteins, or fats, and without which this whole electron transport chain will cease to exist. So far we know that nucleotides are the mononucleotides and they can link with each other through phosphodiester bond, linking the sugar moiety of the one mononucleotide with the another sugar moiety, and the bond between them is linked by the phosphate at 5' and 3', called as the phosphodiester linkage. However, in existence, they usually exist in monomeric form in the solution. And in the solution, the mononucleotides can convert into a dinucleotide or a trinucleotide with the help of a kinase reaction. The mononucleotide just needs an addition of another phosphate to the already existing phosphate, resulting the formation of another bond, the phosphoanhydride bond, and making and converting the mononucleotide adenosine monophosphate to adenosine diphosphate, like for example this here, as you can see in the picture. And in the second reaction also, the diphosphate can change into a triphosphate by the linkage of another bond. So the triphosphate differs from the monophosphate in having three different phosphates attached to it. The first and the foremost, which is present in the 5 prime carbon of the sugar, is called as the alpha phosphate. Then the second one is called as beta and the gamma. So there are actually three different linkages in the triphosphate form of the nucleotide. In nature also, there are certain nucleotides which are in cyclic form, specifically the cyclic AMP and cyclic GMP. Both are needed for the signal transduction of certain hormones as second messengers within the cell to mediate the function of the hormones and to bring out the change in the cell. So the overall summary of the lecture will be like this in the picture. The purines are of two different types present in the nucleic acids, and the pyrimidines are of two different types. They can link with the sugar moiety, form adenosine or deoxyadenosine, similarly for the guanosine or deoxyguanosine. And then when they link with the phosphate, they make adenylate or guanylate or deoxyguanylate. And finally, when they link with each other, and they result in the formation of RNA or DNA. So the RNA will contain adenine, guanine, cytosine, and uracil, whereas the DNA will contain thymine, cytosine, guanine, and adenine as the basis of nitrogen in its polymerization form. So this is pretty much it. You can visit my Amazon page to look through my books and order them, present in both Kindle and paperback format for you. And do subscribe to my channel for further videos which I will upload in due time.